So my tag says Juan. My real name is Juan Pablo, but I dropped the Juan very long time ago when I was a kid because Anglo-Saxon called me Juan and French called me Juan, which means playing. So I kept the Pablo now. Okay. So thank you very much for being able to participate here. So I'll talk about phages in Mycobacterium abscessus, and this work is really run by John Hauer in my lab. And Mycobacterium abscessus, as many of you already know, is a rather difficult to treat infection. Treatment is about 1 to 2 years, and strains are mostly resistant. They're intrinsically resistant, so it creates a lot of problems for treatment. And one thing that we often overlook is that you have two morphotypes, the smooth and the rough. And the rough is much harder to treat than the smooth morphotype. And this will be more clear. So most of the infections we see in Asia happen to be pulmonary infection. These are non-cystic fibrosis patients. In the West, most of the report of Mycobacterium abscessus infection, pulmonary infection, are in cystic fibrosis patients. This is not the case in Asia, even though there's a very significant number of cases of MAPS, pulmonary infection in the region. The other one is mainly associated to plastic surgery, piercing, and so on. And again, even though we see a lot of MAPS in Korea, which is famous for plastic surgery, you hardly ever see any MAPS associated with plastic surgery. It's always almost invariably pulmonary infections. So within the MAPS group, you have 3 subgroups. You have abscessus, sensu strictus, Bolletti, and Massiliensis. They're very similar. One thing that's very important is that abscessus is intrinsically resistant to macrolides. And this is really important because macrolides is the main means of treatment for abscessus. So when I talk about rough and smooth, you can appreciate these are just simple colony from a plate, rough and smooth. And it is believed that only the smooth form can be transmitted from patient to patient, that the rough form is not transmitted. And when we originally get infected with the smooth form, eventually at one point they start to change morphology towards the rough form. From an evolutionary point of view, I'm always puzzled about that, because if you get infected with the rough form and it's the dead end for the bacteria, it just doesn't make sense from an evolutionary point of view. So there has to be some way of returning to smooth from rough. The other thing, nobody has ever isolated a rough form from the environment. The only thing you find in the environment is the smooth form. And I mention this because it will become more relevant during the talk. So again, this is the smooth, the rough. You see it very clearly on the plate. In the patient or in an animal model, the rough form, these serpentine forms. You see this in the Petri dish as well if you look it under the microscope. And now we know that this is due to mutation in this pathway, which is responsible for the synthesis of glycopeptidolipids. So the system is well understood. Now, coming to phage therapy, many of you must be familiar with this paper from Hartford's group. They use 3 different phages, including an engineered, genetically modified phages, to treat a cystic fibrosis patient infected with MAPS infection. So one of the many interesting things about this study is that all 3 phages target the rough form. And in fact, almost all, not almost, absolutely all of the studies where phages have been used to treat Mycobacterium abscessus infection, they target the rough form and not the smooth form. And if I recall correctly, when last I heard Hartford's present, I think they have about 20 or 30,000 different Mycobacterium phages. So it's an enormous collection, but they have just a handful of phages that actually target the smooth morphotype. It's very difficult to find a phage that target the smooth morphotype, which is puzzling. So this becomes even more relevant for us in Singapore, but when we look at strains from Thailand, from Korea, from Malaysia, it's the same. We started by acquiring about 200 strains from the National University Hospital, and we plated them and looked for smooth and rough, and we found is that actually over 50% of them are of the smooth morphotype. So if we're going to be treating patients in Singapore or in Asia, we need phages that target the smooth morphotype as well as the rough. And a percentage is mixed. In reality, there's no proof of this, but we're pretty convinced that most patients carry a mixture of smooth and rough, but it's just a matter of ratio. So when we say we have 50% that are smooth, we don't see any rough on the plates, but if we plate 10 to the 8 organism, probably will find some rough as well, and vice versa, is what we assume. So having that in mind, what we did, we took isogenic strains. So we took strain from a patient, we plated them, we picked the rough, and we picked the smooth, and we used those samples to screen for phages. And here's an example. These are smooth, these are rough. They are the same strain, except one is smooth, one is rough. We did whole genomic sequencing. We know what the genetic differences are. And this is an example using a laboratory strain. You have the rough you see because it clamps, you see the texture, and you see a clinical isolate. This is a phage against the laboratory and the clinical isolates. So that's the strategy we used, and we characterize them, we sequence them, etc., etc. So here becomes the interesting part of the story, is that whenever we do any type of work with phages, the first thing we do is to select for phage-resistant mutant, because that's the best way to understand how the phages work. And to our surprise, when we use phages and we select for resistant mutant, phage-resistant mutant, we see rough morphotype appear. This is our smooth, this is untreated, they remain smooth. When we treat them with phage and select for resistant, we get some very clear rough morphotype. And we've tried this with different strain, different phages, different combination. It's completely reproducible. And I'll say on average, it's about 50% of the resistance will be rough and 50% will be smooth. So this is controlled and treated, they remain smooth. So I'm talking about smooth now. We start with smooth. We treat the smooth, about 50% remain smooth, they're resistant. And out of this, some are lysogen, some have a mutation. In fact, we've realized most of the lysogens also have a mutation. And likewise, with the rough, we have the same phenomenon. So this is what they look like. You can clearly see they're very different. We pick them, we sequence them, and we see here that the one that target the smooth, we've identified they have a similar mutation. These are different strains. We get the same mutation on a glycosyltransferase. And then we go back from different experiments. We pick random resistant smooth mutants. We do PCR for the target gene, and we confirm that we pick up the same mutation. We did a reverse genetic. When I say we, it's Chun Hao and the other guys in the lab. And they did a reverse genetic, and as you can appreciate here, it's resistant to the phage. After the reverse genetic, is susceptible again. And when we use alpha-4 to predict the structure of the protein, we see most of the mutations are frame shift or stop gains on the second domain of the protein. So it makes sense that there is not an active protein. And as expected, when we look at the rough mutants now, they have a mutation in the genes associated with the synthesis, the biosynthesis of the GPL, which is a little bit what we expected. We also did find a few mutants that did not have a mutation in this cassette of genes, but had some other mutation, and we're trying to understand how those work. So as I mentioned, also, most of our mutants are lysogens. So I apologize, this says phage treated. These are these are all the resistant mutant we picked. So for example, here out of after 120 hours, if we pick, we get a 40% mutant, of which 30% are lysogens. But what this doesn't tell you is that out of those lysogen, the majority also have a mutation on one of the genes I just showed. So which I find very interesting and surprising. This is the same with the clinical isolate, just to confirm what we do with a laboratory strain and a clinical strain matches. So then the natural question is, are we seeing an artifact, a laboratory artifact? You know, we get a lot of things in the Petri dish that we never see in the clinic or elsewhere. So we repeat the experiment using zebrafish model, this in collaboration with Laurent Kramer at Montpellier, a collaborator. And here we infect it with the smooth morphotype. We treat it with the phage, and then after 4 days post-infection, we recover all the mutants. And you can appreciate all by one are clearly rough. One of them is neither smooth or rough. It's kind of in between the road. You know, you always find surprising. That's what's fun doing this. And then we did thin layer chromatography to see how does the lipid composition. So you have the control and treated. This is your GPL. The second one is the guy that's in the middle of the road. You see some effect on the synthesis of the GPL, but not complete. That was, you know, the first picture I showed. And for the other one, we see a loss of the GPL. Interestingly, the ones in blue is a transcriptional regulator and not the same mutation we had found in vitro. However, we now know this transcriptional regulator regulates that part of that pathway. And in fact, the other mutants are all the same that we see in vitro. So basically, what we observe in vitro is validated by what we see with the zebrafish in vivo. Likewise, we did it on mice, just using 5 mice. We infect the mice with the smooth morphotypes strain. We treat 3 times, 6 hours, 1 day, 2 days. At 72 hours, terminate the experiment. And again, the resistance, we pick everything that survived. And again, they're overwhelmingly of the rough morphotype and so on. And we see a decrease in bacterial load. However, the mouse model is not a predictive model for Mycobacterium abscessus infection, so we cannot overinterpret the results. So, summary of this first part is this transition from smooth to rough. I really want to emphasize this because it's very significant. If we're going to argue that we want to treat the patients with phages targeting the smooth morphotype, let's say in Singapore, and they end up with the rough morphotype, we've just created a much bigger problem than we started with. So it's something we really have to keep in mind and be well aware. Then we identify this GTF as a possible target. And what we expect, the rough mutants have mutation in the expected gene set, and is reproducible in zebrafish and in mice. And we have a lot of lysogens. And the one thing we haven't done, the big limitation that we have presently in our work, is that we have not managed to make a recombinant phage, unlike Hatful, to make the phage strictly lytic. And in fact, I don't have any slide, but I'll mention it because I think it's important. We realized that we managed to do a knockout and remove the regulator, but if there's any prophage in the Mycobacterium abscessus, that prophage will produce the same regulator and compensate for whatever we've knocked out. So knocking it out is not sufficient, and we've learned that the hard way. Anyway, so we went on and we took the same mutants now that I showed before, and we tested for new phages, and now we see that phages that originally didn't preferentially target the smooth can now target the resistant one, which are rough. And in fact, from an evolutionary point of view, I find this fascinating. Phages that bacteria that hardly have any susceptibility to any phages. Once they become rough, they become highly susceptible to a large collection of phages. So before we had maybe 3, 4, 5, and suddenly we can plate 50 different phages, and the bacteria is susceptible to all of them. So by doing that, we try to combine them again, taking the mutants. We screen for smooth mutant and rough mutant, and we pick a phage and we try to combine them. And if you use this system, it looks very clean. Unfortunately, we use a broth system, it doesn't look as clean. Again, this is primarily due to lysogeny. And we repeat this with a clinical strain. And I'm almost out of time. And I think your talk was very appropriate. And there's no doubt we see the same thing every time we do this with any bacteria and phages. When we combine the phages with a drug, we tend to be much more efficient. And in this case, it's quite dramatic because our phage hardly has any effect by itself, even in combination, even when we have 3 phages together. But when we combine it with a macrolide, we have a very significant effect. And again, this is using clinical isolate, the smooth morphotype, and using the rough morphotype. So we see that the combination with the antibiotic is really, really important. So the summary for this part is that so once they become rough, the bacteria become susceptible to other phages, to a large number of phages. And we could come up with a layer approach trying to combine phages that target the resistant and the smooth. And combination with drug obviously seems to be essential. I know it's really romantic to have a picture, especially in Asia, in the jungle collecting phages, but at the end of the day, all of our phages come from the sewage system in Singapore or in Thailand. And I think I've already gone through the conclusion, so I just want to thank the team. We take a lot of high school students in the lab that come and do rotation from high school, and they really are the ones that screen for most of the phages. And the team, which have a great team working on phages and our collaborators. So thank you. I'll stop there. And time, I think.